Hello, this is Raghu Sai, and this is going to be a tutorial video on AP Biology Concepts 7.9 and 7.10, and these cover phylogeny and speciation. So some important vocabulary terms that you should know for phylogeny are phylogeny, of course, phylogenetic trees, and cladogram. So phylogeny is the history of the evolution of a species or group. And this is usually in relationship to a broad group of organisms and their line of descent. Phylogenetic trees are a branching tree-like diagram that reflect evolutionary relationships. In these diagrams, the branch lengths represent relative genetic change or are proportional to time. So basically, if you look at a phylogenetic tree, you can tell the evolutionary distance between different groups. A cladogram is a type of phylogenetic tree that is also used to illustrate evolutionary relationships, but cladograms don't give a sense of evolutionary change or time change. So the branches in cladograms are purely arbitrary and they don't mean anything. So this right here is an image of a cladogram in a phylogenetic tree to give you a better understanding. So you can see in the phylogenetic tree, there's a time scale. It shows the evolutionary distance. You can get a better understanding of where these groups fall in history. But in a cladogram, these branch lengths, they don't mean anything. They don't represent evolutionary time. They're just there to show where they come in history. So this right here, this is a group because they all come from this recent common ancestor, and this is also a group. So if you take one of these groups, this is called a clade. So a group of organisms that come from one common ancestor is a clade. So this is a clade. These two are a clade. This whole thing is a clade. So yeah. So why is phylogeny important? Well, phylogeny allows us to compare species in terms of their evolutionary history, and phylogenetic trees give a visual representation of the time scale and evolutionary pathway that is occurring. So by having a visual, scientists can better make inferences and it's more tangible for people to understand. But how do scientists make these phylogenetic relationships? How do scientists know? Well, they can know through a lot of means, but one way is by comparing DNA and RNA sequences. So the more similar these sequences are, the more recently the organisms shared a common ancestor. They can also compare amino acid sequences of specific proteins, and the more similar they are, the more similar the gene sequence is, and thus more recently the organisms shared a common ancestor. The third way is they can compare morphology and shared derived traits. So the more traits the groups share, the more recent they shared a common ancestor. And this can also be deciphered using fossil evidence. And one thing that's really important about these relationships and for your understanding is that these are never complete. Scientists are always revising their findings. Scientists are always getting new evidence so they can make better inferences and update their diagrams. So these are not set in stone at all. This right here is a diagram that will help me outline some points that will help you better analyze phylogenetic trees and cladograms. So the first thing that's really important is analyzing the branch lengths. So first identify if this is a cladogram or a phylogenetic tree, because if it's a cladogram, the branch lengths don't mean anything really. And if it's a phylogenetic tree, the branch length can reflect evolutionary distance, so that can be really important when you're answering questions. This right here, this is a junction where in the evolutionary time the taxon split, and this is the most recent common ancestor for A and B. This is a node. The nodes are points of divergence for a possible common ancestor that may have given rise to two or more new taxa. So these are the taxa, A and B. And the reason discussing taxa is because many questions that you'll see about individual organisms often involve searching for a taxa on a diagram. So you have to know which taxa you're looking for. And a taxa is just a biological ranking for an organism. This right here, you might be wondering, is an outgroup. An outgroup is a reference group that is used when finding the evolutionary relationships between different organisms under study. These ones that the outgroup is a reference for these two. This is the in-group. And the outgroup is less related to any of these taxa than any of these taxa, or sorry, the taxa in the in-group are more related to each other than any of the taxa are to the outgroup. So this is just purely for reference. And the most recent common ancestor of the in-group, which is right here, you can see by the node, the most recent common ancestor for this in-group is also much more recent than the in-group's common ancestor with the out-group. So basically what that means is these two are much more related to each other than any of them are to the out-group. Another thing that's really important to understand is the idea of a molecular clock. So the molecular clock measures the absolute time of evolutionary change based on observations that some genes and regions of genomes evolve at constant rates. So this allows us to measure the accumulation of mutations and keep track of evolutionary time, even when we're going millions and millions of years into the past. So this is a graph that's a molecular clock. And the way this is created is basically in 3 steps. You find the number of genetic differences. In this case, it's nucleotide substitutions, but it could also be codon sequence differences. It could be a number of things. So you find the number of genetic differences, then you date the evolutionary branch points based on the known points from the fossil record, and then you just graph it. And then you can get a rate at which evolution is occurring. So from this you can see how much evolution is occurring over time, and we've assumed that it's a constant rate based on the data points from the fossil record. The second topic in this video is speciation. So speciation is essentially just the formation of a new species from an existing one. It's just the creation of a new species. And we have to define a species so that you can better understand it. So a species is a population, or it could also be a group of populations that can interbreed, and the organisms that are produced are capable of producing more offspring, and they're viable. So a population of interbreeding organisms capable of producing viable fertile offspring is a species. And there are 2 main types of speciation. Allopatric speciation is when 2 populations become reproductively isolated. And an example of this could be when there's a geographical barrier separating organisms in 2 populations. The second type is sympatric speciation. So this is when individuals in one geographical area are influenced by disruptive selection or mating preferences. And this can cause speciation. And if you don't know what disruptive selection, it's basically when 2 opposing traits are equally favored by nature in that environment. So that can cause sympatric speciation. So the cause of speciation, like we said, was mating preferences, disruptive selection, geographical barriers. But these can be generalized into 2 categories: pre-zygotic barriers and post-zygotic barriers. And you can guess from the name, these happen before fertilization, after. So for pre-zygotic barriers, some examples would include habitat isolation or behavioral isolation. And post-zygotic barriers could be when there's a creation of a hybrid that's not viable and fertile. One example could be the creation of a mule. They may survive, but they won't be fertile. So some takeaways for you from this. Why do scientists care about phylogeny? And are phylogenetic trees set in stone? These are really important questions, and you can pause the video if you want to answer this question. Okay, so why do scientists care about phylogeny? In essence, this phylogeny allows scientists to make evolutionary relationships and get a better understanding of our past. So that's why phylogeny is important. That's why we have the diagrams, is to show our past and show relationships. And are phylogenetic trees set in stone? Absolutely not. Scientists are always getting new evidence. It can come from the fossil record, it can come from DNA sequencing, it can come from anywhere. Scientists are always trying to get new evidence to better understand the past. And these are the references I used to create this. There's a guided notes worksheet that goes with this, so you can follow along. There's also an extension worksheet for more problems. I hope this was helpful. Thank you.